Hi guys, welcome to Mastermed Academy. I hope you guys are all doing really good. If you are new to my channel, then please hit the subscribe button below and press the bell icon to receive future updates on our new and upcoming videos. And don't forget to follow us on Instagram. So today I'm going to be talking about transplantation immunology. Now, this is a very famous celebrity who featured in Modern Family. If you haven't watched Modern Family, then I suggest you guys to watch it. So this is a famous actress and her name is Sarah Hyland and her stage name is Haley. Now, the purpose of me putting this picture here is she was a patient of a dysplastic kidney and she underwent a transplantation. Now, the first time when she inherited the kidney from her father, after a few months it got rejected, and then the second time when she got the transplant from her brother, it went very well and now she's okay. So if you ever want to annotate a celebrity to transplantation, then you can remember her. Now, what are the types of grafts? Now, before we start a graft, we should know some basic terminology, just like learning a language. Now, the first is allograft. Now, allograft is basically when the donor and the recipient, they belong to the same species, right? This is what allograft is. So tissue from a donor of the same species and the recipient is taken, but they are not genetically identical, right? This is allograft. Now the next is autograft. Now autograft is basically, let's say I'm the donor and I am the recipient myself. So let's say if I have an injury, skin injury, and I want to transplant a part of the skin, then the doctor would basically use my, let's say, thigh, and he will transplant the skin from the thigh to my wrist, let's say. So this is autograft. So now the third is isograft. Now isograft is also called syngeneic graft. Here we have tissue graft between two individuals who are genetically identical. Now when I say genetically identical, I think most of you will be thinking about twins, and that's right. We have monozygotic twins, we have dizygotic twins, right? But if you are a twin, then when we have a transplant between the twins, we call it isograft, right? The last is the xenograft, and here we have a tissue graft from a donor of a totally different species. For example, I'm a heart patient and I need a heart transplant. So one way to get a heart is using a pig's heart, right? So using a pig's heart to transplant and put it in my chest, let's say, this is basically xenograft. Xeno, different species. Okay, so I hope you understood the terminology here. So let's talk about transplantation immunology. Now in immunology, let's say in transplantation, we need to know about HLAs. Now HLA stand for human leukocyte antigens, and I have made a separate video about this called adaptive immunity. And if you guys haven't seen adaptive immunity, I would urge you guys to see it because I've briefly explained there. So basically the genes for HLA are located on chromosome number 6. Now HLA MHC is almost the same thing. MHC stands for major histocompatibility complex. We have two types of MHC. We have MHC1 and we have MHC2. MHC1 reacts with the cytotoxic T cells and MHC2 will react with CD4 helper cells, right? Now both class 1 and class 2 HLA genes exhibit enormous polymorphisms, right? So in transplantation, when we are planning for a transplant, we need to basically match the HLA genes between the donor and the recipient. This is the basic job. Now class 1 antigens are found on all nucleated cells except RBC, because RBC don't have nucleus, right? They present antigenic peptides to CD8 positive cells, cytotoxic cells. Class 2 antigens are found on macrophages, genetic cells and B cells, which are antigen presenting cells basically, and they present antigens to CD4 T cells. Now we basically have to match the donor and the recipient HLA locus, and the more you match, the better the result will be, right? So let's say if I have a twin brother, then the chance for rejection is really low, right? So I hope you guys understood the HLA genes and the MHC business here, right? So we need to match the MHC between the donor and the recipient, right? Now this is the structure of MHC class 2 and MHC class 1, in case if you guys don't know, right? Now HLA matching. Now in HLA matching, there are two sets of HLA genes per patient, let's say, right? So I have two sets of HLA genes. One set is from my father and one set is from my mother, right? and siblings have 25% chance of a perfect match. So I have a younger brother, so if he decides to give me his kidney as a transplant donor, then we have a chance of 25% to match. Now the next is types of transplant rejections. The first is acute rejection. Now before we go into the types of rejection, I would like you guys to know something about allorecognition. What is allorecognition? So let me take you guys to the whiteboard and explain. Now let's talk about allorecognition. Now there are two types of allorecognition. The first one is called the direct allorecognition. And let's imagine that we have a donor antigen presenting cell, let's say a macrophage, and it is presenting a blue antigen, the blue dot, through a red MHC molecule. Now the recipient brown cell, which is called the T cell, will react with the antigen presented by the donor APC, and then when it's activated, it will lead to recruitment of T cells, antibodies and neutrophils, which will eventually lead to the rejection of the graft. The second type is the indirect allorecognition. Let's imagine that we have a donor kidney and that has a lot of antigens, and one of them is the red antigen as presented in the figure. Now this time the recipient antigen presenting cell will recognize the antigen, engulf the antigen and process it, right? And then after processing, this antigen will be presented by the self MHC molecule, right? And then the same story, the brown cell will basically recognize the antigen, which is the recipient T cell, and then once activated, the recipient T cell will recruit other cells like T cells, antibodies and neutrophils, leading to rejection. So the difference between direct and indirect is that in direct we have a donor APC that presents the antigen to the recipient T cell, and in indirect we have the recipient APC which recognizes the donor antigen. So what is acute rejection? Now acute rejection basically occurs due to two processes. There is a cellular process and there is a humoral process. Now in cellular process, what is cellular process? So let's say I'm getting a kidney from, let's say, my friend, and this is a kidney transplant, and when I get the kidney, post transplantation the graft will have some type of antigens there, right? So those antigens are foreign to my body. So what will my body do, right? So the answer is, of course, we'll try to fight, we'll try to recognize the antigen and try to get rid of it because it is foreign to us. So that's why this process is initiated by HLA antigens on the graft that will stimulate the recipient CD4 CD8 positive T cells, right? So my CD4 CD8 positive T cells will react against the donor antigens in the graft, right? So this is one way why the rejection occurs. So humoral immunity means B cells, antibodies, and cellular immunity means T cells. So in humoral process, we have antibodies that developed after transplant and they cause vasculitis. Now what is vasculitis? Vasculitis is basically inflammation of a vessel, right? So there will be a vasculitis of graft vessels with lymphocytic infiltrates. Now how to treat this? So you use immunosuppressants to treat this kind of a problem. Now if you decide to do a second allograft, for example, I take the first graft from my friend and I rejected it. So the question is, okay, if you take the second allograft from the same donor, right? So will my body again reject it or will it accept it? The answer is it will reject it again, but this time it will be much faster. Why? Because we already developed a memory response when I had the first kidney transplanted into me, right? So this is a very important word, the memory response or the memory cells. So we develop memory cells when we reject the first time, so that when you receive the same transplant from the same donor, you will reject it very quickly because you have the memory cells and you already have pre-made antibodies, right? The response is quite robust and fast. Now the very important point to remember here is the onset. It takes weeks to months for acute rejection to occur. So this is a cartoon that shows the acute rejection here, and we can clearly see that there is a alloreactive antibody, right? Alloreactive antibody, my antibody, and this is reacting to the antigen. And because of this, we have parenchymal cell damage, interstitial inflammation, and endothelitis or vasculitis, right? So I hope you guys understood the cartoon. Now this is a histological structure of a vessel, and you can clearly see how thick the tunica intima is, right? This is a fibrosed, thickened tunica intima, and you can clearly see that there is no lumen in this vessel. Can you see a lumen here? No, because it is all occluded by the cells, by the lymphocytic infiltrate, like granulocytes, it could be neutrophils, it could be some kind of lymphocytes. So all of them occlude the vessel, and because of this we have a rejection, right? So I hope you understood the histological structure. So let's talk about hyperacute rejection. Now let's imagine my friend again. I'm really sorry for my friend, I'm taking a lot of organs from him. So let's imagine the same friend, and now he wants to give me, let's say he wants to give me another kidney. Okay, now what happens this time is, when the transplant took place, post transplantation the organ got rejected in minutes. Now why did this happen? Why the rejection took place in just a matter of minutes to hours, right? So pre-existing antibodies were already there in my body. That is the reason why as soon as the transplant took place, the preformed antibodies attacked the graft, leading to rejection. So this is a type of type 2 hypersensitivity reaction. Now what is type 2 hypersensitivity reaction? Here we have the preformed antibodies reacting with the donor antigens and forming a complex, right? So once the complex is formed, these complexes activate the complement, they activate other parts of innate immunity. Also they form a thrombus in the vessels and occlude them. Now the occlusion of the vessel, what will it do? It will lead to necrosis, ischemia, right? So ischemia, what is it? What is ischemia? Ischemia is basically there is no good supply of blood towards an organ. Why? Because there is occlusion of the vessel. So this is ischemia, right? Now pre-existing recipient antibodies will react to donor antigens. The ischemia and necrosis due to thrombosis of graft vessels occur, right? Now if this ever happens, you need to remove the graft very quickly, because it's useless to keep the graft, right? So it must be removed immediately, and it could be prevented by cross matching. And the onset, very high yield to know, the onset is minutes to hours or within minutes, right? Now this is a histological slide showing glomerulus post kidney transplant. Now this is the cartoon of the same thing, hyperacute rejection, and here we have the circulating allo antigen specific antibody which made a complex with the antigen, donor antigen, and then that lead to complement activation, endothelial damage, inflammation and thrombosis. So I hope you guys understood the cartoon. The next type of rejection is chronic rejection. Now as the name says, chronic means, let's say it will take how much time? What do you think? Years, maybe hours or months? What do you think? So let's answer this question. Now chronic rejection can take from months to years to develop. So it's a long time business. Here we have both cellular and humoral mechanisms which are active, and let's say the hallmark of this rejection is arteriolosclerosis. Now what is arteriolosclerosis? Now arteriolosclerosis is basically thickening, you can write it down, thickening, hardening and loss of elasticity of the vessel wall. This is arteriolosclerosis. So the vessels become very thick. Now what are the examples of chronic rejection, right? Let's see. It happens in kidney, and that's called chronic graft nephropathy. In heart, that is the accelerated atherosclerosis. In the lung, we call it bronchiolitis obliterans. And the last but not the least, liver, is vanishing bile duct syndrome, right? This is the cartoon showing chronic rejection, and we can clearly see that we have the donor antigen reacting with the APCs, and then that led to the intimal thickening of the smooth muscle wall. There was also a proliferation of the smooth muscle wall, and then all of that led to the vessel occlusion, right? Now this is a histological slide of the arteriolosclerosis that happened in a kidney graft. So this is a chronic rejection, and this is the vessel here, and you can see the thickness, how thick it is. Now this is the summary of the three types of rejection that I told you. And if you just want to remember one pathology, or let's say the hallmark pathology of each type of rejection, then so in hyperacute rejection, what do we have? In hyperacute rejection, we talked about it occurs within minutes, right? And there is, yes, you guessed it right, we have thrombosis. So here we have thrombosis, and that leads to necrosis and ischemia. In acute rejection, we have vasculitis. So the pathology was vasculitis, right? And in chronic rejection, what did we have? Yes, you guessed it right. In chronic rejection, we had arteriolosclerosis. Arteriolosclerosis. So this is, let's say, the pathology, hallmark pathology that you should know about each type of rejection. Now the last slide is basically treatment. So in here, when you have hyperacute rejection, now think logically, would you require some kind of a drug to basically treat the hyperacute rejection? No, because it occurs within minutes. So before you even give the drug, before you even put the drip or something with the drugs, it's not going to work. So the best treatment for hyperacute rejection is just to remove it. This is the best treatment. So remove the transplant. Now, for acute and chronic, you can use immunosuppressive drugs such as corticosteroids. Everybody knows what is a steroid. Calcineurin inhibitors like tacrolimus and cyclosporin. You can use antiproliferatives like azathioprine and mycophenolate mofetil. And the last is mTOR inhibitors, everolimus. You can also use antibody-based treatments such as basiliximab, declizumab, and rituximab.